Good afternoon, everyone. It's my pleasure to be here. Thank you for inviting me. And thank you for giving me the opportunity to talk about our latest work with the O-PTIR. So firstly, I would like to say, I'm gonna skip this part because it's been perfectly covered already by Mustafa. But I just wanna say that I was completely new to the spectroscopy field until less than 2 years ago. I'm a biologist, but as Antti mentioned before, I always focused on nanotechnology and nanomedicine, but never used spectroscopy before. And when I joined Professor Peter Gardner's group, I was immediately fascinated by the opportunities that the infrared and Raman spectroscopy techniques can offer when applied to cell biology. But I was also aware of the disadvantages that they have, of course, but luckily enough, I guess I was in the right place at the right time, which is when Professor Gardner got the new O-PTIR instrument. So I had the chance to get trained by Mustafa and Sergey from Photothermal with the instrument, and I had the chance to get some data out of human cells. This is our setup. So we have, in terms of infrared lasers, we have an OPO laser that, as already mentioned, covers the high wave number region, a QCL laser that covers the lower wave number region, so in particular the fingerprint region. Then, of course, we have a Raman spectrometer. In our case, so the sample is made of cells grown on a calcium fluoride cover slip. And then when ready, another cover slip of calcium fluoride is placed on top of them, and then they're simply placed inside the microscope upside down. Our aim was here to use this O-PTIR technology to then investigate cancer cells, because we are familiar with them to start with. Firstly, in a fixed sample, but then the most challenging aim was to develop a sample preparation and acquisition protocol to study live human cancer cells. So when a buffer or an aqueous buffer was present. So let's start with a panoramic of with the OPO and QCL laser. So here is an optical image of a cancer cell. This is a mIRage cancer cell. mIRage is a pancreatic cancer cell line. Basically what I did, I just took the image and then draw a grid with multiple spots at 1 micron step size from each other. And started an hyperspectral acquisition. I here selected few spectra just to appreciate differences across the cell regions. So to start with the OPO laser, we can clearly identify amide A and amide B bands, which represent the protein content of the cell, and then of course the CH stretching region, which represent the lipid content. Then with the QCL laser, we have, of course, the dominant peak is the amide 1, followed by the amide 2, again, represent the protein content. We have also a lipid peak at 1740. And also other peaks at lower wave number that represent both protein, lipids, and also nucleic acid content. But let's have a look a bit closer to the OPO, the spectra obtained with the OPO laser. You have here the same optical image, but also next to it you have the infrared image of the same cell, taken at 2935. So it is interesting to see that there is a high intensity region in the middle of the cell, more or less, which doesn't correspond with the nucleus, I would say, because the dark spot here, which is most likely the nucleolus, it's far away from this high intensity region. So what we did, we took again a hyperspectral, but in this case I selected few spectra in a line at 1 micron step size between each other again. And as you can appreciate, even only after 1 micron from each other, there is a huge variability in terms of intensity across the sample. In fact, you have the highest intensity spectra, which are the red and the blue, which of course were taken at the higher intensity spots at the top, and then followed by the lower intensity other spectra. In particular, I would like to highlight the olive green spectrum, which has almost no protein content, but it's kind of rich in lipid content. While on the other hand, we have the light green spectrum, which is just 1 micron next to it, but has a quite abundant protein content, but not lipid. So we can think that the olive spectrum was a lipid droplet. But what kind of lipid droplets do we have inside the cells? Well, many, I would say. In particular, if we look at the endoplasmic reticulum that I here marked in green, we can see that covers almost all the cytoplasm of the cell. It can appear in 2 different shapes. It can be as a tubular shape or as a circular shape when the cell is more close to the mitosis stage. And this is interesting because it really looks like the circular feature that we observed before in the IR image, exactly these ones. But also we have Golgi vesicles that are essentially concentrated next to the nucleus often, and we have lysosomes which are also vesicles but are more spread around the cytoplasm, I would say. Of course, the nucleolus, which is the darker spot inside the nucleus, as you can see here, is also present. So let's move on to the OPO, but concomitantly acquired with the Raman. So we have also our favorite olive green spectrum here again, and as you can see, this figure is confirming the high lipid content of this spot. In this case, the spectra are normalized in order to appreciate better the differences between each other. But also it's interesting to see that if we select 3 spectra from the nucleolus, you can see how they are different compared to the other spectra. In this case, we have the green, the purple, and the black. As you can see, the CH2 absorbance, it's much reduced compared to the other ones. This feature was also confirmed by Raman. In fact, there is no peak, although the peak in this case was not so pronounced like in the O-PTIR. This can mean that there is a lack of long-chain lipids in the nucleolus, which makes sense because we are not expecting to find any phospholipids in this region of the cell. Then let's have a look at the QCL, at the data obtained with the QCL lasers from the same regions. If we focus again on the spectra obtained from the nucleolus, the green, the purple, and the black, you can see how the shape and the breadth of the amide 1 peak, it's different. It looks like it's a broader shape of the band, and there is a shift towards higher wave number. Also, the absence of phospholipids is confirmed because there is absence of the peak at 70-40, while in the other spectra is clearly present. So this is confirming that we are in front of nuclear material because we attributed this change of the shape of the amide 1 peak to, rather than an increase in beta sheet, but to increase in DNA and RNA material, because we know that nucleolus is that region of the nucleus where the ribosomes are assembled, and we know that the ribosomes are rich in rRNA. So this result also is very interesting and makes sense. Then let's have a look at the Raman obtained simultaneously from the same spot. What we have here, we have a pronounced peak of the amide 1, and then the amide 2, which is not so pronounced compared to the infrared. We have the amide 3 peak as well, which is also includes protein content, so it's a combination of NH and CH stretch, which is very interesting because we can see there is a lot of variability across sample. Plus we have a peak at 1235, which is the symmetric PO2 stretch, so it tells us something about DNA and RNA content again. And interestingly enough, we can see that the spectra obtained from the nucleolus are also have a higher intensity in this region compared to the other spectra, which confirms what was observed with the infrared. Okay, now let's move on to the even more exciting part, the one about live cells. So in this case, it was not possible to get an optical image because of lack of contrast, but it was possible to get very nice images with infrared. In fact, I acquired here 2 images with 2 different cell lines, again MIA-PACA, and then I added the MDA231, which are a breast cancer cell line, just to compare if there were differences across different cells. The images were acquired at 1550 because I wanted to avoid the water absorbance that is observed at 1640 usually, but it's not at 1550. And it worked. In fact, we can appreciate few regions of high intensity. And I again performed an hyperspectral analysis and selected a few spectra just to check the differences. If you, with the MIA-PACA, with the blue, in the blue spot, which I took from high intensity region, it was possible to observe a nice peak at 1088. Which it's telling us that we are in presence of nuclear material. And this is a very important finding because I wasn't able to see this peak in a fixed sample, but I am able to see it in live cells. So when probably has to do with the fact that DNA, it changes its conformation from A DNA when it's dehydrated to B DNA when it's hydrated. And this is also reported from literature. And this makes, it's very important to make live analysis as well for this reason. Also, I took another spectrum, the purple one, where it was possible to appreciate a big peak in the lipid region, which it's interesting because this spectrum comes from the edge of a cell, I think, which is of course a region that we expect to be rich in phospholipids because it's probably where the plasma membrane is. And then simply with the MDA231, I did exactly the same thing, and everything I saw with the MIA-PACA was also observed with the MDA231, confirming the reproducibility of the technique. Okay, then I carried on observing the MIA-PACA cells, and I took here 2 infrared images of live cells. This time I tried to get exactly the same spots with the 2 different lasers, which is challenging enough because when you switch from the OPO laser to the QCL laser, you have to reinitialize the instrument. So essentially you lose the cell location, although you can save the spots where you are. So in this case, we were able to get basically overlapping images with the two lasers. You can see that this images here is obtained with the QCL laser, and you can see how the brightest spots obtained with the QCL do not necessarily correspond to bright spots obtained with the OPO, which essentially highlights lipids. In this case, for example, if you look at the green spot, the green spectrum, you can see that with the QCL I was able to see a very good peak at 1088, again suggesting that we are observing nuclear material. But on the other hand, there were no lipids, essentially no lipids. So yeah, probably we are observing the nucleus. But if you have a look at the other two spots, the purple and the blue, that also comes from a bright region obtained with the QCL laser, you can see that instead they are not rich in nuclear material, but instead they are rich in lipid material, and this was confirmed also with the OPO laser. If we have a look at also the amide one region, in particular the 1740 peak, you can see how this theory was confirmed. So I took here two spots, two spectra from the green area that before was observed being rich in nuclear material, and you can see that here they don't present the phospholipid peak, but on the other hand, the blue and the purple, they have a nice big peak at 1740, which essentially is confirming what was also observed before, so that in the nucleus there is no phospholipids, and the phospholipids that you can appreciate with the QCL, you can also see with the OPO laser. Okay, so to conclude, this was the first use of the OPO and the QCL O-PTIR lasers and concomitant Raman for spectroscopic analysis of fixed and live cells. This technique made possible to acquire spectra of IR and Raman with a very high spatial resolution at the same time from the same spot. The spectra obtained are pure and present no spatial blur, and we were able to obtain undistorted amide one band in live cells, so in presence of water. So this is a very basic study, but we think it opens the doors to enhanced studies of cells, and in particular, for example, drug-cell interactions, study of cell metabolism, and in general, cell response to external stimuli. With this, I would like to thank all the supervisors, the PIs, Peter, Jane, and Joe, and I would like to thank the Mustafa and Sergey that trained me from Photothermal, the founders from AstraZeneca as well, and of course, thank you for listening.
Thank you very much, Alice, for your really interesting presentation. There was a question, I think you were also covering it afterwards, but I was just thinking, do you have some other things to add? So using the Raman and O-PTIR, is it possible to identify the lipids with distinct spectra? So I think you were also covering it a lot, but do you have some special things to add on that?
Well, I don't know what to add, because I think I already covered the fact that, yes, lipid have different features, but in particular from here you can see, you can focus on the stretch CH2 versus CH3. So you can do also a ratio between these two the CH2 and CH3, so you get information about what kind of lipid it is.
Yeah, good. Another question is that was there a reason why the liquid cell was placed upside down?
Okay, well, this is will not sound very scientific, but we simply noticed that in this way we got very nice spectra, very nice, and with a very little water absorption. I'm not sure if Mustafa already covered it in his
yes.
talk. Yeah.
Can I actually comment on that just very quickly?
Yeah, sure.
Yeah, it's a good point. The biological cell is grown on the calcium fluoride flat, right? And then because the light with the infrared light and the green light come from the top, if you put the cell on the bottom, a drop of water, and then a cover slip on top, normally the infrared light will have to pass through some finite thickness of water before it reaches the cell and then is detected via the green channel. So by placing it upside down, you now have the cell sitting on the top, hanging upside down, but obviously well adhered. And so the infrared light hits the cell first. When it hits the cell first, it's unattenuated by the water. So you have a much more intense O-PTIR signal from the cell. And then underneath it, in fact, it doesn't matter how much water you have after that, because all of that information is then contained within the green, because the green interacts with the photothermal effect, and the green then passes through. And in your case, you collected the signal in transmission mode, whereas I spoke mostly about reflection. So that's one of the other advantages of having it upside down. You can have a relatively thick passing of water. You can flow things. You don't have to be confined to a typical sort of 7-10 microns that you would with normal FTIR. Yeah, I hope it explains it.
Thank you.
Yeah, thank you. And probably a very general question, which is typically always in life science or very often. How about the evaporation of the water? So could you comment on that? So what was that? How to avoid that in this case?
Yes. So this is a good question. I have a sketch here that I made on how I prepare what we call the sandwich of the samples. So essentially, we noticed that we reduce drastically evaporation, drawing around the cell sample, so at the edge of the calcium fluoride slide, essentially a line with a pap pen, which is basically contains an hydrophobic dye. So making this hydrophobic border, then you avoid water dispersion first, and then once you place your cover slip on top of them, you create a sort of, you squeeze your sample, you create a sort of, not vacuum, but essentially it's a sealed kind of at the edge. So we noticed that even, for example, when we left the sample running overnight, the morning after, the water was already there. So there was no evaporation, the cells were fine. And so that worked.
Auntie, actually, I'll just quickly point out that we have actually developed our own cell, so you don't have to go through this drawing process anymore. There'll also be a flow cell. So you can now, from us, obtain a cell with variable path length spaces in which you can flow liquid as well to keep the cell alive and maybe deliver drugs or other things.
Yeah, this is more homemade, cheap.
Yes.
Yeah, no, but it's actually, I think very many of the great inventions are done exactly this way. So I think that was a very clever. So well done.
Start somewhere, right?
Yeah, indeed, that's true. So thank you very much, Alice. This was really interesting.